Yes. Good morning, students. Today we discuss with the previous year's questions from two chapters, that is microbes for human welfare and biodiversity and its conservation. These two chapters are last chapters in plus 2, but these are also very important chapters. Last few years there will be several questions asked from these two chapters. Then we first start up with the first question from microbes for human welfare. That is the unit last but 3 units, and it is conversion of milk to curd, improve its nutritional value or nutritional quality by increasing the amount of. So what is takes place during the conversion of milk to curd? Milk to curd, there will be a lactobacillus, the bacteria called lactobacillus, lactic acid bacteria, lactobacillus, that will convert milk to curd. It improves the nutritional quality. Nutritional value means nutritional quality. Nutritional quality by increasing the amount of vitamin B12. Vitamin B12, increasing the quality of nutritional quality of vitamin B12, and it is called probiotics. It's called probiotic. That is the first question. That is conversion of milk to curd improves its nutritional quality by increasing the amount of vitamin B12. Vitamin B12. That is the first question. Okay. Then second one. Yes. Identify the microorganism which is responsible for the production of an immunosuppressive molecule, that is cyclosporin A. That is another part in this role of microbes in, that is bioactive molecules. Bioactive molecules, that is called cyclosporin A. Cyclosporin A. It is an immunosuppressive agent. Immunosuppressive agent used in organ transplantation. Organ transplantation. Immunosuppressive agent, that is cyclosporin A. Cyclosporin A obtained from a fungus that is called Trichoderma polysporum. That fungus is called Trichoderma polysporum. You heard about Trichoderma fungus. Trichoderma is applicable in both the two purposes. It is one, it will act as immunosuppressive agent, that is Trichoderma polysporum. A species of Trichoderma polysporum is used as immunosuppressive agent or bioactive molecule, and another species of Trichoderma, it is used as biocontrol agent. Another Trichoderma species is used as a biocontrol agent. Biocontrol agent. Fungus, and it is mainly used for that Trichoderma species, mainly used in treating some plant pathogens, to controlling some plant pathogens. That is the Trichoderma species used for biocontrol agent. Here it is the microorganism which is responsible for the production of immunosuppressive molecule or immunosuppressive agent. Cyclosporin A. Cyclosporin A, it is obtained from the fungus Trichoderma polysporum. Trichoderma polysporum. Okay. And others are, you have Clostridium botulicum. It is mainly the fungus. Clostridium botulicum is a bacteria. It is used for the synthesis of that is the industrial production of butyric acid. Clostridium botulicum is responsible for the synthesis of butyric acid, industrial production of butyric acid. Aspergillus niger, you have heard about the citric acid synthesis. That is citric acid synthesis. And Streptococcus, several species, you have idea about Chinese Streptococcus species. Several Streptococcus pneumoniae here. It will be another species called Cerevisiae. Streptococcus cerevisiae. There are several species of Streptococcus. There will be we get a streptokinase enzyme from the Streptococcus bacteria. We get a streptokinase enzyme, and it is used as a clot buster. It is used as a clot buster to remove the blood clots during myocardial infraction. These are example for these biosynthesis of or synthesis of industrial synthesis of Clostridium butylicum for butyric acid, Aspergillus niger citric acid, and Streptococcus cerevisiae. Streptococcus, there are several species of Streptococcus. Here it will be Streptococcus, it is used in streptokinase, some bacteria, streptokinase species that is used as streptokinase bacteria, streptokinase, an enzyme. Streptokinase enzyme, it is obtained from bacteria called Streptococcus. Okay, then next question. Match column 1 with column 2 and select the correct option using the codes given below. That is also some members produced the commercial products, and here it will be the important microbial organisms. What are the select the correct option? Citric acid, the product. Citric acid, biosynthesis of citric acid by industrial production by the Aspergillus niger. Aspergillus, the species is niger, Aspergillus niger, citric acid. Cyclosporin A, that we discussed in the first question, cyclosporin A, it is obtained from Trichoderma polysporum. Statins are produced from a yeast, Monococcus purpureus. Statin, produced from Monococcus purpureus. A yeast called Monococcus purpureus. It is used for Monococcus purpureus. It is the statins produced from the Monococcus purpureus is used as lowering the blood cholesterol. Lowering the blood cholesterol level. Blood cholesterol. Statins, a yeast, it is produced from Monococcus purpureus. It is mainly used for lowering the blood cholesterol. It is another bioactive molecule. Then butyric acid. Butyric acid is industrially produced from Clostridium butylicum. The bacteria called Clostridium butylicum. That is butyric acid. Then citric acid, Aspergillus. Cyclosporin A, Trichoderma polysporum. Then statins, Monococcus purpureus, and butyric acid, Clostridium butylicum. And option is box. Okay. Yes. Then next question. Which of the following is wrongly matched in the given table? Wrongly matched. Micro product and its application or users. Or what is the function? Streptococcus, from which the product will be streptokinase. It is genetically engineered, modified by genetically engineering. It is modified to produce the enzyme. Back from the bacteria Streptococcus, streptokinase enzyme is produced, and it is used as removal of blood clots. Clot from blood vessel. That is clot buster. It is called streptokinase, is otherwise called clot buster. This is used for treating the patients that are suffering from myocardial infraction leading to heart attack. That is the first one, Streptococcus, and it is correctly matched pair. Micro product and application. Second one is Clostridium butylicum. That is lipase. The product is not lipase, it is butyric acid. Not lipase, butyric acid. Lipase produced from other fungus, that is Candida species. Okay, Clostridium butylicum, it is butyric acid, and it is the commercial synthesis or industrial synthesis of butyric acid from Clostridium butylicum. And it is not remove oil stains. It is lipases that will remove oil stains. That is the wrong match. It will be Clostridium butylicum. It is not produce lipases. It is produces butyric acid, and it is commercial or industrial production of butyric acid, not removal of oil stains. Oil stains are mainly used as lipases. Lipases not produced from the Clostridium butylicum. It is from a fungus. Okay. Then third one, Trichoderma polysporum. That is cyclosporin A. Correct. And it is an immunosuppressive drug that we already discussed. Trichoderma polysporum. Cyclosporin A. Immunosuppressive agent. Then last one, Monoscus purpureus. Monoscus purpureus, that is produce the statins. Statins, lowering of blood cholesterol. Lowering of blood cholesterol level, that is statins. That is a yeast called Monoscus purpureus. It is called Monoscus purpureus. It will produce some statins, that is lowering the blood cholesterol level. And the wrongly matched one is answer box. The option is box. Wrongly matched. Okay. The next one. Monoscus purpureus is a yeast used commercially in the production of. That is also we discussed what is the purpose of Monoscus purpureus. That is, some statins are produced. Some statins are produced and it is used for blood cholesterol lowering agent. It is considered as blood cholesterol lowering agent. And the option is the. Okay. Then others are ethanol. It is by yeast, Streptococcus, Saccharomyces cerevisiae, ethanol. Ethanol is produced from Saccharomyces yeast. Saccharomyces bread, that's called ethanol, yeast. Saccharomyces cerevisiae. Saccharomyces cerevisiae, ethanol. Then streptokinase for removing clot bloods, from clots from the blood, and it is called clot buster. It is called clot buster. This is produced from Streptococcus bacteria. Streptococcus bacteria. Then citric acid, Aspergillus niger. Aspergillus niger. Commercially produced from the fungus. Aspergillus niger. Aspergillus niger. And here Monoscus purpureus. That is a yeast called Monoscus purpureus is a yeast used commercially in the blood cholesterol lowering statins. Blood cholesterol lowering statins. Okay. Then next one. A patient brought to a hospital with a myocardial infarction is normally immediately given. What is given? A patient brought to a hospital with a myocardial infarction is normally immediately given what? Clot buster. And clot buster is streptokinase. Streptokinase. It is obtained from Streptococcus. It is called a clot buster. It is called a clot buster. Remove the blood clots, the blood vessels. Blood clots are removed by clot buster streptokinase. Immediately given. That is the case of streptokinase. Okay. Then next one. Read the following statement having two blanks, A and B. There are two, fill up the blanks, A and B. A drug used for dash patients is obtained from A. Obtained from a species of organism B. What is the matching? Two blanks. The matching of two blanks, that is a drug used for organ transplant patient. Organ transplant patients. That is organ transplant. A drug used for organ transplant, that is cyclosporin A. That drug is cyclosporin A. Cyclosporin A. A drug used for cyclosporin A. Patients is obtained from a species of the organism Trichoderma. Trichoderma fungus. Polysporum. The species is called Polysporum. Trichoderma polysporum. The option is box. That is a drug used for organ transplant patients is obtained from a species of the organism is Trichoderma polysporum. The option is box. Okay. Then next one. Which one of the following is the wrong matching of a microbe and its industrial product, while the remaining three are correct? So which one? Based on these four options given, which one is the wrong matching of a microbe and its industrial product, while the remaining three are correct? Yeast, a yeast variety that is produced statins from Monoscus, that yeast called Monoscus purpureus. Monoscus purpureus will produce statins. Okay. Acetobacter aceti, that is produces acetic acid. Commercial production or industrial production of acetic acid from Acetobacter aceti. Another bacteria called Acetobacter acetyl. Acetic acid is synthesized. Vinegar, the main content of vinegar, acetic acid. Then Clostridium butylicum, it is commercial production or industrial production of not lactic acid, it is butyric acid. Not lactic acid, Clostridium butylicum is for the industrial production of butyric acid. Not lactic acid. Lactic acid by lactobacillus. Lactic acid bacteria that is called lactobacillus. Here it is butyric acid. This is the wrong matching. Wrong matching is the Clostridium butylicum that will produce not lactic acid, it produces butyric acid. Then Aspergillus niger, that is citric acid. The commercial or industrial production of citric acid from the fungus. The fungus called Aspergillus niger. That fungus is very important, Aspergillus niger, and it is coming under Aspergillus niger is coming under the fungus class group, which is the class group Ascomycetes. You have already studied in the fungal part. That is fungus called Aspergillus. There are several species of Aspergillus. Aspergillus is coming under the fungal classification. It is called Ascomycetes, and it is used for commercial production or industrial production of citric acid synthesis. And the option is third one is the wrong matching, that is Clostridium butylicum, it is lactic acid. Okay, then next question. Which one of the following pairs is wrongly matched? Again, the wrong match. Wrongly matched. The alcohol is, that is the ethanol, ethyl alcohol is produced not by the enzyme nitrogenase, it is during alcoholic fermentation by yeast. The enzyme is called zymase. The enzyme is called zymase, not a nitrogenase. That is the zymase. Alcohol which is produced during this alcoholic fermentation using yeast. It is alcohol produced from the yeast using zymase. That is called enzyme called zymase, not nitrogenase. That is the wrongly matched one. Then you have note about fruit juice. What are the enzymes involved? The preparation of the fruit juice, commercially prepared, or that is in the case of the bottled fruit juices. Bottled fruit juices that is we purchase in shops are very clear due to the enzyme it is used for clearing or clarification by two enzymes, that is called pectinase and proteases. Proteases and pectinases. Clarification or clearing agent used in bottled fruit juices that will become more clear. The bottled fruit juices are more clear. In our home, it will be more turbid. It will be more clear using these two enzymes called proteases and pectinases. The fruit juices, the bottled fruit juices were more clear, and it is called clarification. That process is called clarification. Fruit juices are cleared or clarified. Fruit juices are cleared or clarified by the enzyme pectinase. That is correct. Pectinase and protease both are involved. Then textile, it is the enzyme is amylase. Amylase is involved in textile industry, food preparations. They are all needed. This textile also, the amylase enzyme is involved. Then detergents that is lipases. That is detergent formulation, removing the oily stains. Oily stains. Oily stains. It is removed by using this lipase enzyme or hydrolyzed by lipase enzyme, removing the oily stains, and it is mainly involved in detergent formulation. The detergent formulation that is made by, and it is mainly applicable in detergents are mainly laundry. It is mainly applied in laundries. Laundries are an application. That is in the case of detergent, lipases. The lipases are involved in detergent formulation. Detergent amylases used in textiles. Then pectinases and proteases used for clarification of fruit juices. And here thymase enzyme is needed. That is in the case of alcohol making from yeast. That is alcohol fermentation. That is enzyme is produced with thymase, not a nitrogenous. The answer is A. Wrongly matched pair is A. Okay. Then next one. Which one of the following is used to manufacture ethanol from starch? Ethanol means what? Ethyl alcohol. What is the organ used to manufacture ethanol from starch? That is the what is the bio that is in the case of organism or microbe involved is Saccharomyces cerevisiae, or it is called yeast. Saccharomyces, that is yeast. Saccharomyces, alcohol synthesis from starch, that is by the Saccharomyces cerevisiae. Okay. Then next question. Which one of the following microorganism is used for production of citric acid in industries? Citric acid production is by Aspergillus niger, that we already discussed in the last questions, match the following, etc. Is production of citric acid, biosynthesis or citric acid synthesis, that is by Aspergillus niger, that is a fungus. Aspergillus niger is a fungus that belongs to the class Ascomycetes. Rhizopus is another fungus. Rhizopus, bread mold you have heard about. Then lactobacillus, lactic acid bacteria, and penicillium. There will be penicillin is obtained from Penicillium notatum. Another species called notatum, it is Penicillium citrinum. Okay. So here the citric acid synthesis is from Aspergillus niger. These are the industrial products, that is, or industrial acids, mainly industrial synthesis of acid. It is mainly from some microbial organisms, by some old biotechnology techniques. The strain as self it is used itself, that the strains as it is itself it is used. Strains as itself is used for preparing these acids. That is the old biotechnology method. It is called the strains as such is used. As such is used. Okay. But that is very important data in the role of microbes. Role of microbes in human welfare. The synthesis, this acid synthesis, industrially produced. Question next. The citric acid is also another question. The citric acid is produced by the Aspergillus niger. Aspergillus niger. That is another repeated question in 1995. Aspergillus niger. Okay. Then next one. Which of the following is put into anaerobic sludge digester for further sewage treatment? Further sewage treatment. But that next role of microbes in sewage treatment. Sewage treatment consists of two steps. Sewage treatment consists of two steps. First is called primary treatment and second is called secondary treatment. Primary and secondary treatment. And what will take place during primary treatment? That is the physical process. Primary treatment, what happens? Physical process, that is filtration and sediment. That is removal of solid waste, solid substances, suspended solids. They are all removed. Filtration and sedimentation. Filtration and sedimentation. And the fractions, that is gritted, that is called soil, small pebbles, they will be sedimented or sedimentation. That is called first process called primary treatment. And that after primary sedimentation of this solid waste, etc., or removal of solid debris, etc., and filtration and sedimentation, the primary effluent, the supernatant produced above, that primary effluent is passed through the secondary treatment plant. And the secondary treatment plant is the process is called biological process. Primary treatment is physical process and the secondary treatment is biological process. During this process, what happens during secondary treatment? The biological organisms, that is called microbes, are growing in this large aeration tanks. And it is mechanically agitated. That large aeration tanks, that will be mechanically agitated. That primary effluent is primarily mechanically agitated, and pump air. Air is pumped into it. That is aeration tanks, and the tanks are called aeration tanks. And microbes are growing, and microbes will oxidize these organic substances in the waste, sewage or primary effluents, and it will reduce the BOD. BOD means biochemical oxygen demand. It will reduce the BOD level and the water is more clear. And that is, after that sediment is called that is sediment, that will be the activated sludge. Activated sludge, that is in the form of bacteria in associated with the fungal filaments to form the activated sludge. It is sedimented and that in a settling tank. And these sediments, activated sludge is transferred to or pumped into another tank. It is called anaerobic sludge digester. It is called anaerobic sludge digester. That is, activated sludge is put into anaerobic sludge digester. That is during secondary treatment. There are two processes. What are the two processes? During secondary treatment, that is both aerobic process. First aerobic process, and second is anaerobic process. Anaerobic process. First aerobic and second is anaerobic. And in first aerobic bacteria, that will oxidize all the organic substances in the primary effluent, and all these primary sludge will be sedimented in the settling tank, and that sludge will be activated sludge. That sludge is called activated sludge. Activated sludge will transfer to another tank. It is called anaerobic sludge digester. Anaerobic sludge digester, where there will be anaerobic process with some anaerobic organisms. The presence of some anaerobic bacteria. Anaerobic organism, that is anaerobic bacteria, it will produce some biogases. That is contain methane, carbon dioxide and hydrogen sulfide. Methane, hydrogen sulfide and carbon dioxide. That is the process of sewage treatment. Sewage treatment plant, STP, that is called STP. Sewage treatment plant. Sewage treatment. For sewage treatment, we have both the primary treatment and a secondary treatment. Primary treatment is a physical process. Secondary treatment is that is a biological process, in which first aerobic bacterium that will grow and it will oxidize all the organic substances and reduce the BOD biochemical oxygen demand or biological oxygen demand, reduce the BOD. And next is the activated sludge. After the secondary effluent is removed and the sediment is called activated sludge. That is activated form of bacteria. Activated sludge, it will be settled and effluent is passed to the water bodies. After significantly reducing the BOD level, then the effluent is transferred to water bodies without any pollution. And the sludge, activated sludge in the sedimenting tank or a settling tank, that will transfer to active anaerobic sludge digester. And for further sewage treatment, here some anaerobic process will take place. Anaerobic sludge digester, anaerobic process, it will produce some bacteria, anaerobic bacteria and fungi that will produce this biogas and biogas production. And biogas mainly contains methane and some traces of hydrogen sulfide and carbon dioxide. Okay. Now here, which of the following is put into anaerobic sludge digester for further sewage treatment? That is the activated sludge. It is the active. From the activated sludge, what it produces? The biogas. By process called anaerobic process, and bacteria is called anaerobic bacteria. And main biogas is methane, and methane producing bacteria, they are generally called, these anaerobic bacteriums are coming under methanogens. That bacteriums are called methanogens, producing methane. Predominant gas found in this anaerobic sludge digestion is methane, and that bacteriums are called methane producing. That is called methanogens. That anaerobic bacterium present in the anaerobic process, and it is present in activated sludge. Activated sludge, it consists of several anaerobic organisms or especially anaerobic bacteria, and it are generally called methanogens. And important genera belong under methanogen is called methanobacterium. It is called methanobacterium. That will reduce the substances convert into biogas. Biogases. Biogas production. Okay. Here, which of the following is put into anaerobic sludge digester for further sewage treatment is activated sludge, and that treatment is anaerobic process. Further process is anaerobic process by anaerobic bacteria. That is very important role. Very important role of microbes in sewage treatment. Sewage treatment, that is both aerobic role and as well as anaerobic role. Okay. Then next question. Which one of the following is in sewage treatment removes suspended solids? Suspended solids. What is removed? The suspended solids are removed by which method? Which of the following sewage treatment removes suspended? That is the first process. That is first process. It is called primary treatment. Primary treatment, and it is a physical process. It is a physical process. Physical process. That is filtration and sedimentation. Removal of solid waste or suspended solids. For suspended debris, they are removed. That's by filtration. Sometimes it will be filtration, and further sedimentation. The solid particles like that is a soil, clay, there is small pebbles, etc. And that small soil particles and pebbles, they are called, small pebbles are called grit. That fraction is called grit. G-R-I-T, grit. That fraction is called grit. Soil and small pebbles. Soil and small pebbles. They will be sedimented. Suspended solids. That is during primary treatment. Sewage treatment, first process, that is primary treatment, and it is a physical process. Physical process. Okay. Another treatment is secondary treatment, and it is a biological process. Clear? Then next. What gases are produced in anaerobic sludge digesters? During anaerobic sludge digestion from this activated sludge, what will be produced? What are the gases it is produced? And generally that gases are called biogas. That gas is called biogas. Biogas produced by anaerobic sludge digester, and they are methane, hydrogen sulfide, and carbon dioxide. The predominant gas is methane. The predominant gas that is produced during this anaerobic sludge digestion, and it produces methane, then hydrogen sulfide, traces of hydrogen sulfide, and carbon dioxide, and they are collectively called, these three gases are collectively called biogas. Biogas production. For biogas production, that we have studied another heading in this chapter, that is role of microbes in biogas production. That is next heading. Next heading of this chapter, and before that we studied this in this part of this chapter, that is anaerobic sludge digestion. For sewage, in sewage treatment plant, we produce where we get methane, hydrogen sulfide, and carbon dioxide. That is collectively called biogases. Okay. Then next question. The domestic sewage in large cities. What is sewage? That is municipal waste. Wastewater. Municipal wastewater. That is domestic sewage. Domestic sewage in large cities contains is processed. It contains several microbial organs and is highly contaminated, and VOD value is very high. Then is processed. The domestic sewage in large cities is processed by aerobic and then anaerobic bacteria in the secondary treatment in sewage treatment plants. Sewage treatment plant is called STPs. STP means a sewage treatment plant. So in the domestic sewage in large cities is processed. During this process is called a sewage treatment plant. Sewage treatment plant. Here it is processed by aerobic and then anaerobic process, and secondary treatment takes place during sewage treatment plants, STP. It is called a sewage treatment plant is called a STP. That is the process. So after that, primary and secondary treatment, the effluent from secondary treatment, that will be, or the cleared water or unpolluted water is transported to water bodies or passed to water bodies. That is the very important point in domestic sewage in large cities. It is processed by both aerobic and anaerobic bacteria. Aerobic process and anaerobic process by aerobic bacteria and anaerobic bacteria in the secondary treatment. That treatment is called secondary treatment, and it is a biological process. Secondary treatment is a biological process in sewage treatment. Sewage treatment plant is called STPs. It is called STPs. Okay. Then next one. Secondary treatment is mainly a. What is the secondary treatment? Secondary sewage treatment. Secondary sewage treatment is a biological process. Primary treatment is physical process. Primary treatment is physical process, and the secondary treatment is biological process. And the primary treatment is filtration and sedimentation. And the secondary treatment consists of both the aerobic and anaerobic process. Aerobic and anaerobic process. Involvement of both the organisms, aerobic organisms and anaerobic organisms. Okay. Then next one. Which one of the following pairs is wrongly matched? Another pair that is wrongly matched pair. The yeast. Yeast is mainly used for the production of ethanol by alcohol fermentation. Ethanol production from yeast. By using yeast, ethanol is produced. That is correct one. Then another one is streptomycetes. Streptomycetes is an antibiotic. That is the bacteria that will produce streptomycin. It's called produce an antibiotic called streptomycin. Antibiotic called streptomycin, not the streptococcus. Streptococcus produces an enzyme called streptokinase. Enzyme is called strepto. Here it is not streptococcus, it is streptomycetes. Streptomycetes. This is another type of bacteria, streptomycetes. It is coming under streptomycetales. And it is another group of bacteria. Not it is eubacteria. It is another type of bacteria. It is coming under streptomycetales. It is called strepto. And from this, an antibiotic called streptomycin. Streptomycin is produced from this bacteria, streptomycetes. That is also correct one. Then coliform bacteria, that is the E. coli, Escherichia coli, coliform bacteria, that is Escherichia coli, it is not produces vinegar. Vinegar is produced by the acetic vinegar or acetic acid. Acetic acid is the main content of vinegar, and here it is the bacteria called Acetobacter aceti, not a coliform bacteria. It is Acetobacter aceti, will produces the acetic acid. Commercial production of or industrial production of acetic acid. Commercial production of acetic acid. That is also your chapter part, that is called the role of microbes in the production of industrial production of acids. Industrial production of acid. We have studied already butyric acid, lactic acid, citric acid, and another one is called acetic acid. Acetic acid is industrially produced from the bacteria called Acetobacter aceti, not the coliform bacteria. That is wrong. Not is produced coliforms, not produce the vinegar or acetic acid. That is a wrong statement. And methanogens are producing the methane producing bacteria or biogas. That is producing the gobar gas. Gobar gas is a biogas. Gobar gas is a biogas. It is produced from gobar. Gobar is obtained from cattle excreta. That is cow dung, etc. Cow dung is called gobar. And using this cattle excreta or cow dung, we produce this anaerobic digestion, and it will produce this gobar gas or biogas. Gobar gas is also called biogas. We loaded this, that is in the case of the biogas plant. Biogas plant with the cattle excreta or cow dung with water, and it will produce the gobar gas by bacteria called methanogens. Methanogens are methane producing bacteria. Methane producing bacteria, that is called methanogens. This gobar gas is also produced by methanogens, methanobacterium or methanogens. Gobar gas. Gobar gas is the biogas. It is produced from gobar. Gobar is cattle excreta or cow dung, especially that is it will produce. Okay. Here the wrong pair is the coliforms never produce the vinegar. Coliform never produce the vinegar. Then option is, it will be C. Option C. Okay. Then next one. Which bacteria is utilized in gobar gas plant? That is we have already discussed. That is called methanogens. Methanogens, methane producing, or this gobar gas mainly contains methane, and also carbon dioxide. Methane and carbon dioxide, main gases, and also traces of sulfur, etc. Then next one. Gobar gas mainly contains methane and carbon dioxide. Okay, methane and carbon dioxide. Then next one. Which of the following can be used as a biocontrol agent in the treatment of plants? But now we go through this next role of microbes, that is called role of microbes in biocontrol agents. And there are several roles for bacteria, virus, and fungi. They all act as biocontrol agent, controlling several diseases. Here, in the case of the biocontrol agent, it is used as the treatment for plant diseases, treating the plant diseases, or plant to control the plant pathogens. That is very important member is called trichoderma fungus. It is a root-borne fungus. It is called root-borne ecosystem. Root-borne ecosystem where the fungus are trichoderma species. Trichoderma species that is used as biocontrol agent for controlling the plant. But trichoderma is a fungus species, and it is a root-borne fungus. It is mainly a root-borne fungus, and it is mostly controlling the causative for plant pathogens, especially the plant pathogens, trichoderma. And the trichoderma, where another species we have studied in this bioactive molecule. That is called cyclosporin A. Cyclosporin A, that is also produced from the fungus called what? It is called Trichoderma polysporum. Here it is, trichoderma is used as biocontrol agent. Both trichoderma have both role in the case of human welfare. That is in the case of trichoderma. Some trichoderma species will act as biocontrol agent, and other ones are worked as this biocontrol agent, and another one is bioactive molecule as well as biocontrol. And a microbial organism that is involved in both bioactive molecule as well as biocontrol agent is called trichoderma. Okay. Then next one. Chlorella you have studied in green algae. Chlorella is a green algae member. Unicellular green algae. Lactobacillus you have already known. Lactic acid bacteria. Lactobacillus. Anabina is a cyanobacteria. Free living cyanobacteria. Anabina, nostoc anabina, etc. are cyanobacteria. Okay. Then next one. Select the correct group of biocontrol. What are the correct group of biocontrol? A list of biocontrol agents. That is studied. That question is frequently asked questions in NEET examination. In one paper, that is the year after or year before, last one. That is last before. That is called a question. That is 2 times it is asked. That is the biocontrol agents. In same paper there are 2 places it is asked. That is first one. Here it is trichoderma. It is a fungus used as biocontrol agent in controlling plant diseases. And another microbial organism for biocontrol is a virus called baculovirus. That virus is called baculovirus. A genera coming under baculovirus is called nucleopolyhedrovirus. What we called NPV. NPV is nucleopolyhedrovirus. Baculovirus. And what is the feature of this baculovirus? Baculovirus are species specific and narrow spectrum. Species specific and a narrow spectrum. That will control only the targeted members, targeted species. Not all the species, some targeted species. Especially this baculovirus will control arthropods or insects. and it is control only the targeted insects only, not target species, non-target species. There are only target insects. It will control only target insects. It will control only target, and it is called narrow spectrum. It is called a narrow spectrum, species specific baculoviruses. That is it is mainly promoted by IPM, Integrated Pest Management. IPM is supported this baculoviruses as biocontrol agent. They will enhance or support. Then third one is bacteria. It is Bacillus thuringiensis. Bacillus thuringiensis, what we commonly called BT. Commonly called BT. You heard about BT cotton, BT BT brinjal, etc. Bacillus thuringiensis. It is mostly controlled this insect, that is butterfly caterpillars. Mostly that is the case of Bacillus thuringiensis, BT toxin. Here the three members for biocontrol agents: Trichoderma, baculoviruses, and Bacillus thuringiensis. Bacillus thuringiensis, what we called BT. Okay, so these are the three biocontrol agents in your syllabus. Very important. There is a fungus and a virus and a bacteria. Bacillus is bacteria. So fungus that will control plant diseases are called Trichoderma species, and baculovirus coming under the nucleopolyhedrovirus, that is a genera coming under that general group called baculoviruses, that is species specific and narrow spectrum. And it is another one, bacteria, it is called Bacillus thuringiensis. It will control these butterfly caterpillars. It is only control that, which is Bacillus thuringiensis. Okay, yes, next one. Microbe used for biocontrol of pest butterfly caterpillar is, that we already discussed. Microbe used for biocontrol of pest butterfly caterpillars. It is by Bacillus thuringiensis. Bacillus BT. This BT, Bacillus thuringiensis. Over here it is controlled by this a sacket of the spores obtained from this BT, Bacillus thuringiensis, a sacket of spores of this bacteria, Bacillus thuringiensis, mixed in water and spraying on the vulnerable plants. Vulnerable plants, it will spray on the vulnerable plants, and the insects will eat the leaves of the plants, and in the gut of this the larvae, it will release the toxins and it will kills them, and that population is removed. Okay, that is controlling of this BT, and now it is used as BT is genetically engineered, and the genes for this BT toxin is incorporated in plants, and it is called BT cotton, BT incorporated cotton, that is BT cotton, BT brinjal, etc. Okay, but Bacillus thuringiensis. Next one. A nitrogen microbe associated with Azolla in rice fields. Our next part is the last part of this chapter, that is microbes for human welfare. That is the role of microbes in biofertilizers. Before that, biocontrol. Here it is biofertilizer. The role of microbes in biofertilizers. We have known about several biofertilizers using these microbial organisms. Here, a nitrogen-fixing microbe. Nitrogen-fixing microbe associated with the Azolla. Azolla is a fern. Water fern. Azolla is a water fern, and it is considered as the smallest fern. Azolla, and sometimes it is called Azolla is called goddess of fertility. It is called goddess of fertility. Azolla. By nitrogen-fixing microbe associated with the Azolla. Azolla is a fern, that is a fern, that is pteridophyte. Pteridophyte member, the leaves of this Azolla, water fern. It is called a fern, that is a water fern, and it is the smallest fern, and it is sometimes called goddess of fertility. In associated with the leaves of this Azolla in rice fields is an cyanobacteria called Anabina. That cyanobacteria called Anabina. Then is association between Anabina with the Azolla leaves that will synthesize nitrogen fixing. It will produce nitrogen fixing, nitrogen fixing. And it is in rice fields. The fertility of rice fertility is increased by Azolla with the Anabina association. Anabina association. That is a very important association. Nitrogen fixing microbe associated with the Azolla. But here the nitrogen fixing microbe is Anabina. It belongs to cyanobacteria, free living cyanobacteria. Free living cyanobacteria, that is Anabina. Anabina is associated with the Azolla. Azolla leaves in rice fields. Increases the nitrogen fixation. Increase in the nitrogen fixation. That is very important. Azolla with the Anabina. Anabina is a cyanobacteria. Blue-green algae. Cyanobacteria is sometimes called blue-green algae. Blue-green algae. Then, which one of the following microbes forms symbiotic association with the plants and helps them in their nutrition? What is the organism that's or microbe symbiotic association with plants and helps them in their nutrition? That is symbiotic association. Symbiotic association, that is by mycorrhiza. Mycorrhiza, association of fungus with roots of higher plants, that is called a mycorrhiza. And here the member mycorrhiza, the fungal partner will be glomus. The fungal partner of mycorrhiza is called a glomus. Fungus. The fungus is called a glomus. Fungal partner. Fungal partner, that is called a glomus in mycorrhiza. Symbiotic association and helps them in their nutrition. That is mycorrhiza. Okay? Mycorrhiza. Yes. Next one. Which one of the following helps in absorption of phosphorus from soil by the plants? That is also mycorrhiza. Absorption of phosphorus or mobilization of nutrients. Phosphorus mobilization from the soil by a fungus, that is a fungal partner that is present in mycorrhiza. Mycorrhiza, that is in the case of the fungal partner, is called glomus, that will absorb, or that is in the case of the flow of absorption of minerals, especially phosphorus from soil by mycorrhiza. Main function of mycorrhiza is the absorption or transport or mobilization of phosphorus from soil. Okay. The next one. An organism used as a biofertilizer for raising soybean crop. Soybean belongs to leguminous plant. Leguminous plant. Pyrotherial varnana soybean. Soybean is most proteinaceous, most protein. Soybean, leguminous plant. In leguminous plant, there will be an organism used as biofertilizer. That is what it is called, a bacterium called a rhizobium. Nitrogen fixation done in leguminous plants by rhizobium. Rhizobium bacteria. Rhizobium bacteria in association with the roots of these leguminous plants and forms nodule. Root nodule formation. Root nodule formation, and it will fix the nitrogen. So rhizobium is the bacteria involved in the leguminous plants. Leguminous plants, the nitrogen fixation is done by rhizobium bacteria. Okay, the next one. Nitrogen fixation in root nodules of ulnus is brought about by. Ulnus is a non-leguminous plant. Non-leguminous plant. Ulnus. In non-leguminous plants, the nitrogen fixation is carried out by another bacteria. It is called Frangia. That bacteria is called Frangia. And it is also not a eubacteria. Frangia belongs to a class called Actinomycetes. Actinomycetes is the connecting between bacteria and fungi. Actinomycetes. That class is called Frangia. Frangia bacteria coming under the Frangia strain of bacteria that is coming under Actinomycetes. Actinomycetes. It is called rare fungi. Sometimes it is called Actinomycetes. Members are called rare fungi or branching bacteria or rare bacteria, etc. Okay, that is called ramified bacteria, rare fungi, etc. Frangia. About the non-leguminous plants like ulnus, casuarina, ulnus, etc. These plants, the nitrogen fixation is carried out by not rhizobium, it is by bacteria called Frangia. Non-leguminous plants, the nitrogen fixation is carried out by Frangia. Okay, the next one. Which of the following species do not have ability to fix atmospheric nitrogen? Which organism is not able to fix atmospheric nitrogen? Acetobacter, it is a free-living nitrogen-fixing bacteria, aerobic bacteria. Anabina and nostoc are also free-living cyanobacteria that will fix atmospheric nitrogen. And Spirogyra is a green algae. It has never fixed the nitrogen. green algae. Spirogyra belongs to green algae. It is called pond silk. Spirogyra. It is belongs to filamentous green algae. Filamentous that you have studied in plant kingdom. Plant kingdom, that is the green algae. Chlorophyceae members. Spirogyra. It cannot fix the atmospheric nitrogen. Other three, Acetobacter, Anabina, Nostoc, they are all fix the atmospheric nitrogen. Spirogyra cannot fix the atmospheric nitrogen. Okay. Next one. Which one is one of the following statement is correct? Legumes fix nitrogen only through. Yeah. Which one of the following statement is correct? Legumes fix nitrogen only through the specialized bacteria that we called rhizobium that live in the roots. Legumes fix the nitrogen. Legumes fix the nitrogen. Nitrogen fixation by leguminous plants. Leguminous plants only through the specialized bacteria that is called rhizobium. That bacteria is called rhizobium in the roots. Symbiotic association between rhizobium and the roots of leguminous plants and forming root nodule formation, and it will fix the atmospheric nitrogen. Option is A. Then question number next. Now we go to next chapter, that is the biodiversity and conservation. Previous questions are mainly involved. Previous year questions that is involved in this both these chapters, microbes for human welfare as well as the biodiversity and its conservation. We hear the PYQ, that is previous year questions from the biodiversity and its conservation. Okay, the first one, among the able quartet, which one is considered the most important course driving the extinction of species? What is the able quartet? The able quartet is the four major courses of biodiversity laws. Able quartet is four major courses of biodiversity laws. Biodiversity laws of biodiversity. Four major courses of biodiversity laws, that is called able quartet. It is called able quartet. What are the four major courses of biodiversity laws? That is first one, habitat loss and fragmentation. Second, overexploitation. Third, alien species invasion. And fourth one is co-extinction. There are four major courses. Out of which one is considered as most important course? Out of these four courses of biodiversity laws, which one is the most important course driving the extinction of species? Driving the extinction of species. Which one is the important course driving the extinction? That is habitat loss and fragmentation. That is the important course or main driving force. Main driving course that is driving the extinction of a species. Extinction. The species extinction is mainly by habitat loss and fragmentation. These are called able quartets. Able quartet. Four major courses of biodiversity losses. Okay. Then second one, which of the following regions of globe exhibits or exhibits the world, the globe or world exhibits highest species diversity or greatest species diversity? Greatest species diversity, it is Amazon, the tropical rainforest of Amazon, Amazonian rainforest. Amazonian rainforest is the highest or greatest source of species diversity. Species diversity, it is called Amazon forest. Lungs of the planet. It is called lungs of planet. Lungs of the planet. Lungs of planet. That is called Amazon rainforest. That is greatest biodiversity. So there are 12 mega biodiversity centers in world. Greatest 12 mega biodiversity centers, and include one. India is also one among the mega biodiversity centers in the world. Out of this, the greatest one is the Amazon rainforest, and it is called lungs of planet. Okay, next one. According to Robert May, the global species diversity is about, and the species diversity, global level, the global species diversity is studied by several scientists and several agencies, IUCN, etc. And the most scientific and conservative and scientifically sound data is from Robert May. According to Robert May, the global species diversity, GSD, global species diversity is 7 million organisms are. According to Robert May, that is so far it is reported will be 7 million. According to Robert May, it is most most conservative and scientifically sound method by Robert May is GSD. Global species diversity is it is 7 million organisms are reported so far. 7 million organisms. Okay. Then, and in this chapter there will be several data regarding this species diversity, or in the case of this biodiversity conservation. All these things there will be several data. IUCN data. There are several data are important, and several years and several agencies or several summit or protocols or some that's called summit or conferences. They are all very important. Then next question: decline in the population of Indian native fishes. Indian native fishes due to introduction. About decline, there will be decline in the population of Indian native fishes due to the introduction of Clarius gariepinus. What is Clarius gariepinus? Clarius gariepinus is African catfish. The scientific name of African catfish. African catfish is called Clarius gariepinus. It is introduced illegally in India. Illegally it is introduced in India for aquaculture. What aquaculture, that is the case of Clarius gariepinus is illegally introduced in India. That will leads to the decline in the or extinction in the population of Indian native fishes, that is Indian catfishes, native catfishes. Indian native catfish due to the introduction of Clarius gariepinus, that is African catfish. But after introducing this African catfish, Clarius gariepinus, that will lead to the extinction of our indigenous means Indian native fishes, catfishes, due to in Yamuna can be categorized as, which one is? Here it is alien species invasion. Alien means foreign or exotic. Alien species or introduced exotic species or foreign species. Exotic or foreign species. Alien species invasion. Alien species invasion. That is one of the cause of biodiversity loss. That is, first one is example African catfish. And there are several introductions or introducing several organisms in some countries that will lead to the extinction of their indigenous species. That you heard about Nile perch. Nile perch introduced in the Lake Victoria in East Africa. That will extinct several species of their indigenous cichlid species, cichlid fishes. There are several data in there. Okay, over here it is alien species invasion. An example, that is African catfish introduced in India. That is Indian native species. That will decline in the population of Indian native fishes or extinction of Indian fishes. That is by alien species invasion. Okay, then next. Alexander von Humboldt described for the first time. Who is Alexander von Humboldt? You heard about Alexander von Humboldt. Alexander von Humboldt is a German naturalist. German naturalist and a geographer. German naturalist and Alexander von Humboldt. And he explored these jungles of South American jungles, and he explained the species area relationship. First time he will explain the species, how these species, number of species related to area, the relation between species and area. That we studied the data or the graph. You have studied a rectangular hyperbola. That is very important. Study by Alexander, that we get a rectangular hyperbola. Species rich, that is species richness, and that is in the case of area and species richness. Number of species. And the arrow will be rectangular hyperbola, and it is converted into a log scale. And here the species, number of species is equal to CZ. Say the power Z. And in the log scale, it is the log scale. Here log S is equal to log S, the species, number of species, log S is equal to log C plus Z log A. The two equations are very important of Alexander von Humboldt. German naturalist and a geographer. You are exploring the area of South American jungles. And he studied the relation between species with the area. And it is the data is given. Yes. Then next one. The organization which publishes the red list of species is IUCN. International Union for Conservation of Nature and Natural Resources. The full name of IUCN. We abbreviate the IUCN. IUCN is the short form and abbreviate IUCN. International Union for Conservation of Nature and Natural Resources. Red list published by IUCN. ICFR means Indian Council for Forest Research Education. Indian Council for Forestry Research Education. UNEP is United Nations Environment Program. United Nations Environment Program. WWF, all of them are know WWF, Worldwide Fund for Nature. IUCN will give the red list data. Okay, that is very easy question. The next one. It is very important, that is in the case of this pie diagram explains the global species diversity of invertebrates. This pie diagram explains this global species diversity of invertebrates. Here the symbols given below is the representation of the extent of global diversity of invertebrates. What groups of 4 portions of this pie diagram A, B, C, D represent respectively? What are they? A, for this data of global species diversity of invertebrates. Here A for insects. Insects. First one A is insects. Then B is what? A is insects and B is not crustaceans, mollusk. B is mollusk. C is crustaceans. And D is other animal. The 4 portions of this pie diagram of global species diversity of invertebrates. Over here, there are 3 pie diagrams in your book, that is global species diversity. First is vertebrates, invertebrates, and third plants. 3 pie diagrams are given. Here the pie diagram, first A is insects, B is mollusks, C is crustaceans, and D is other animal groups, and the option is D. That pie diagrams are also very important. Then next. Which of the following represent maximum number of species among global species diversity? Which among the following in the plant data or the global species diversity of plants or the pie diagram? It explains which of the following represent maximum number of species. That is only fungi. Maximum. Mosses and ferns are very little. Algae is very small part. And lichens are very small part. Very small portion. That is most important is fungi. Maximum number. The next question. Which of the following has maximum genetic diversity in India? There are 3 types of diversity you have studied. Genetic diversity, species diversity, and third one is community diversity or ecosystem diversity. Here, which of the following has maximum genetic diversity? Maximum genetic diversity in India is rice. There are 50,000 strains of genetically different strains. 50,000 different genetically different strains of rice will be produced in India. That is in the case of maximum genetic diversity. 50,000 genetically different strains of rice varieties are put, and it will be rice. And 1,000 varieties of mango. That is in the case of the another one. Maximum is rice. 50,000 strains of rice varieties. Okay. Then next. Which organization published the Red Data book? Red Data book again we studied, that is in the case of IUCN. International Union of Conservation of Nature and Natural Resources. IUCN, the full name is International Union of Conservation of Nature and Natural Resources. The UNEP you have already discussed in the last question, United Nations Environment Program. Then WWF is Worldwide Fund for Nature. Global, that is Environment Facility, GF. GF means Global Environment Facility. Global Environment Facility, GEF, Global Environment Facility. That is the funding agency for environmental studies, environmental conservation, environment funding agency for Global Environment Facility, GEF. Okay. Then next one. Which one of the following have the highest number of species in nature? Which one is the highest number? Here it is in the case of insects. In the total organisms reported so far, in the mostly the animals, 70% of the all total organisms in the earth are 70% it will be animals. 70% will be total organisms. 70% animals. And less than 22% will be the plants, including fungi. Including fungi, algae, bacteria, algae, bryophyte, teratophyte, gymnosperms, all these things below less than. That will be less than 22%. Less than 22% are plants. Total majority of the organisms, 70% of the organisms are animals. And out of these animals, total animals reported, 70% will be insects. 70% will be insects. Out of 10 animals studied, out of these 10 animals, 7 were insects. That is 70%. 7 are insects. That is the most, which one is following have the highest number of species in nature, it will be the insects. Okay. Then next. Biodiversity of a geographical region represent. Biodiversity of geographical region. What is biodiversity of a region, geographical region? That is the diversity of organisms. The diversity means what? Heterogeneity. Heterogeneity or diversity. Heterogeneity or totality of organisms. Totality of organisms living in a region. That is a geographical region. Geography. Geography means distribution of a biological organism. That is a geography. Distribution of geography region. By a diversity, the diversity or a heterogeneity, variation, heterogeneity in the organism living in the region, it is called biodiversity. That is the definition, it is called biodiversity. Okay, then next. Which one of the following pairs of organisms are exotic species? Exotic species means alien species. Alien. A-L-I-E-N. Alien species. In the textbook it is given alien species. Not exotic species, it is called alien species. Alien species is also called exotic species, or it is called introduced species or foreign species, etc. Introduced in India. India introduced exotic species. Which one of the following pairs of organisms introduced in India? Introduced in India. Alien species. That is, one is called Lantana camara, a plant called Lantana camara, and another is called water hyacinth. Water hyacinth. Water hyacinth is called Eichhornia. Eichhornia crassips. C-R-A-S-S-I-P-S. Crassips. Eichhornia. Water hyacinth. Lantana camara. Water hyacinth. They are introduced species or exotic or alien species. And another ones are also in your syllabus. You heard about the carrot grass. Another introduced species, Parthenium. It is called Parthenium. It is called Parthenium. Parthenium is another introduced species in India. It is contaminated through wheat. It is contaminated through wheat seeds. Wheat seeds is Parthenium. And another introduced or alien species, foreign species, Lantana camara, water hyacinth, Parthenium, carrot grass. Parthenium is also called carrot grass. Carrot grass is also introduced or alien species invasion. Alien species invasion. Okay. Then next one. The historic convention on biological diversity, the Earth Summit, was held in Rio de Janeiro in the year. The historic convention on biological diversity, the Earth Summit, Earth Summit was held in Rio de Janeiro, Brazil. Rio de Janeiro, Brazil, in the year 1992. It is in the year 1992. Earth Summit is summit held in Rio de Janeiro in the year 1992. Earth Summit is also called UNCED, United Nations United Nations Council for Environment. Council for Environment and Development. United Nations Council for Environment. United Nations Conference. Conference. United Nations Conference for Environment and Development. Environment and Development. UNCERT. It's called United Nations Conference for Environment and Development. UNCERT. Earth Summit. Earth Summit was held in Rio de Janeiro, 1992. To conserve the main aim will be to conserve the biodiversity. Okay. Then, which of the following is not a method of ex situ conservation? What is ex situ conservation? Which of the following is there are two types of biodiversity conservation, that is called in situ and ex situ. In situ conservation is called on-site. It's called on-site conservation. That is protecting all forest. To save a tiger, all forest is conserved. That's on the site it is conserved. On the total site or total habitat it will be conserved, and it is called on-site, or it is called in situ. It is called in situ. Another one is called ex situ conservation. Ex situ, that is called off-site. Off-site conservation. That is, some of the organisms or it will be critically endangered, or it will be extinct in the near future. That will be conserved by the special care in some special settings with the humans. That is called ex situ, or it is called off-site. Zoos, botanical gardens, wildlife safari parks, etc. And then next, in the case of cryopreservation, then IVF, all these methods, then tissue culture, micropropagation, they are all methods. It is used as ex situ conservation. That is, the animals or organisms that are protected by humankind, human care, special care in some special settings. That is out of their natural habitat, that will be taken from their natural habitat, and it is protected in special settings. It is called ex situ conservation. Here, in the case of which of the following is not a method of ex situ conservation? We hear in situ conservation. In India, they are national parks, wildlife sanctuaries, biosphere reserves, and sacred forest. Sacred forest or groves. Sacred forest or groves. Covered sacred forest. But national parks, wildlife sanctuaries, biosphere reserves, sacred forest. Ex situ conservation, we have studied that zoo, zoological, botanical garden, then in the case of this wildlife safari parks, etc., they are all belongs to this ex situ conservation. But each of the following is not a method of ex situ. In vitro fertilization is an ex situ conservation. National park is not an ex situ conservation, it is an in situ conservation. In situ. Micropropagation is an micropropagation, that is tissue culture. The principle behind tissue culture, micropropagation, tissue culture, plants, that is a ex situ conservation. And cryopreservation, preserving the gametes, preserving the gametes of organisms in a very low temperature, minus 196 degrees centigrade in liquid nitrogen, storing in liquid nitrogen. That is cryopreservation. The cryopreservation, micropropagation, in vitro fertilization, they are all belongs to ex situ. National park is an in situ conservation. Option is box. Okay, next one. In situ conservation refers to the protect and conserve the whole ecosystem. To save the tiger, the total forest is conserved. To save the tiger is tiger save ചെയ്യണമെങ്കിൽ അതിനെ എവിടെ ഇട്ട് സേവ് ചെയ്യണം? ആ സൈറ്റിൽ ഇട്ട് തന്നെ സേവ് ചെയ്യണം. അല്ലെങ്കിൽ ദാറ്റ് ഹാബിറ്ററ്റിൽ, ഫോറസ്റ്റിൽ ഇറ്റ് inhabit in our forest, as such it is it will be protected or conserved, and it is called in situ protect and conserve the world ecosystem. That is called in situ conservation. Next one, the Earth Summit held in Rio de Janeiro in 1992 was called for what? What is the main aim of this Earth Summit? Earth Summit, the main aim will be for conservation of biodiversity and sustainable utilization of its benefits. Conservation of biodiversity, main aim of the Earth Summit. Earth Summit is to conserve the biodiversity and sustainable utilization of its benefits. That is the main aim of Earth Summit held in Rio de Janeiro in the year 1992. Okay. Which of the one of the following is not a method of in situ conservation of biodiversity? Which one of the following is not a method of in situ conservation of biodiversity? Here, sacred groves or sacred forest is an in situ conservation of biosphere reserve. It's also in situ conservation. Wildlife sanctuary is also in situ conservation, and botanical garden is a ex situ conservation. Off-site, ex situ conservation, or it is off-site botanical garden. Others are wildlife sanctuary, sacred forest, then biosphere reserve, national parks. They are all in situ conservation. Okay, the next. Western Ghats have a large number of plant and animal species that are not found anywhere else. So in Western Ghats, there are large group of number of plants and animals that are not found anywhere else. Not found anywhere else. That is, that species will confine to only special location. That is Western Ghat population. Which one of the following terms will you use to notify such a species? That some species are localized in some special regions and nowhere it will be seen. They are called endemic species. Endemic. And that process is called endemism. It is called endemism. Endemic species. It is called endemism. That is a very important question in IISER NIT in 2019. Okay, Western Ghats have a large number of plant and animals that are not found anywhere else, and which of the following terms will you notify such a species? That is called endemic species. Endemism. Endemic. Localized on special regions. Nowhere it will be seen. Keystone species, that is a species in a ecosystem. They are highly influencing and their number will be very limited. The organs are very limited and their influence is very large in that ecosystem. And that organisms are called keystone species. That is, we call the key species or keystone species. Okay, that you studied in the ecosystem. Which one of the following is related to ex situ conservation of threatened animals and plants? What is the method or following conservation? Ex situ conservation. Biodiversity hotspots are in situ. What is hotspot concept? What is hotspot? Hotspot concept was given by Norman Mayers. And what is the concept? Biodiversity concept. That is in the case of the place where the highest number of species, species richness, the highest species richness, and that is the greatest degree of endemism. Species richness and endemism is very high in the places called hotspots, biodiversity hotspots. That's called biodiversity hotspots. Initially in the world there are 25 hotspots. Now 9 are added, and presently there will be 34 biodiversity hotspots all over the world. Biodiversity hotspots. Okay. Then Amazon rainforest, hotspots, Himalayan region. These three are in situ conservations. Wildlife safari park is the ex situ conservation. Wildlife safari park is only the ex situ conservation. Ex situ conservation of threatened animals and plants. So which one of the following is related to ex situ conservation of threatened animals and plants? That is only wildlife safari parks. Biodiversity hotspots, Amazon rainforest, Himalayan region, they are all in situ conservation methods. Okay. Next one. The region of biosphere reserve, which is legally protected and where no human activity is allowed to known as. For biosphere reserve, the structure of the biosphere reserve is 3 parts. That is in the case of, there are 3 regions of biodiversity, biosphere reserve. The most outer region is called transition zone. This zone is called transition zone. Inner to that it will be the buffer zone, and innermost zone is called core zone. Innermost, most interior is called the core zone. This is the structure of a biosphere reserve, BR, biosphere reserve. Here the transition zone, here human settlements. There are human settlements are seen in the case of transition zone. Human settlements. Human settlements are seen in the case of this transition zone. And in the buffer, over here there will be several human interrelation relations, or humans are mostly settled in the transition zone. Then buffer zone, here only scientific research and study purpose are used in only buffer zone. It is only allowed scientific research, study trips, etc. are good up to buffer zone. And core zone is highly legally protected. Core zone is legally interior. Interior will be legally protected, and no human activity is allowed. No human encroachment. No human encroachment. Core region core region is highly interior of the forest. Interior. And that core zone is the biosphere reserve, the region, innermost region. Interior region is called core zone. It is highly protected, legally protected, and where no human activity is allowed. No human activity is allowed, and that zone is called a core zone. And the structure of this biosphere reserve was given by MAB, Man and Biosphere Program of UNESCO. Man and Biosphere Program of UNESCO. UNESCO is program on MAB. MAB means Man and Biosphere. Man and Biosphere Program of UNESCO. It will give the structure of this biosphere reserve. Biosphere reserve in the structure MAB. Man and Biosphere Program. Okay. Then next one. How many hotspots of biodiversity in the world have been identified till date by Norman Mairs? Till date means present. The data will be 34. 34 biodiversity hotspots. Now up to date, it will be 34. 34 hotspots are recorded by Norman Mairs till date. Initially there will be 25. Initially 25 and 9 are added. Okay, the next one. The species confined to a particular region and not found elsewhere is termed as endemic species. That we already studied in the Western Ghats. Some species of Western Ghats are not seen nowhere. And it is called endemism. It is called endemism. Alien means foreign species or exotic species. Keystone species already we discussed. Then, in which of the following both pairs have correct combination? What are the pairs correct? On Institute Conservation National Park, Institute Conservation Botanical Garden, and the option is C is correct. Institute. Here, seed bank is wrong. Seed bank is the Institute Conservation. Then Institute National Park is an Institute Conservation. That is wrong. Then Institute Conservation not tissue culture. Tissue culture is Institute, and the Sacred Grove is Institute. That is wrong. Institute Conservation National Park and Institute Conservation Botanical Garden. That is correct. And here cryopreservation is Institute and Wildlife Sanctuary is Institute. So here answer is only C. Correct combination is C. Then next. Which one of the following areas in India is the hotspot of biodiversity? Western Ghats. Western Ghats and Sri Lanka, Indo-Burma and Himalayas are three hotspots in the India. India is important hotspots of biodiversity in India. Western Ghats. Western Ghats is a hotspot one among the India. One among the hotspots. Out of 34. Out of 34 Western Ghats in India. India there are three hotspots. One of these Western Ghats. Then sacred groves are especially useful in sacred groves or sacred forest. In the sacred forest, all the animals and trees, wild animals and trees are venerated, and it will be highly protected. Venerated and it gives highly protected and conserving the rare and threatened species. They will conserve the rare and threatened species in sacred groves. Sacred groves or sacred forest. Sacred groves or sacred forest. We called sacred groves. Conserving rare and threatened species. Here, all in this sacred groves, all these animals and plants are venerated. They are venerated and they give special care. They give special care and conserving their rare and threatened species. Okay. And there are several sacred groves we seen in the India. That is mostly the Khasi and Jainsha hills in Meghalaya, Aravali hills in Rajasthan. Aravali hills in Rajasthan. They are all examples of the sacred forest or sacred groves. Okay, now this time World Summit on Sustainable Development 2002 was held in South Africa, Johannesburg. The World Summit on Sustainable Development in the year 2002 it was held in South Africa, Johannesburg in South Africa, and it is a follow-up conference of Earth Summit. Follow-up conference of Earth Summit, that World Summit on Sustainable Development, South Africa, Johannesburg in the year 2002, and 190 countries are pledged. 190 countries are pledged to control the biodiversity loss at 2012 highly significant level. Conserve the biodiversity or control the biodiversity loss at a high significant level up to in the year 2010. 2010 at a global level, regional level, and local levels. That is the main aim of World Summit. World Summit on Sustainable Development. It is conducted in Johannesburg, South Africa, 2002. Okay. Then next one. Which of the following is considered as hotspot biodiversity in India? That we already covered. That is Western Ghats. Western Ghats is one of the hotspot of India, biodiversity hotspot in India. Okay. The next one, Biodiversity Act of India was passed in the Parliament in the year 2002. The same year, the World Summit on Sustainable Development conducted in Johannesburg, that is in the case of South Africa. Here, after that, India Biodiversity Act of India was passed by the Parliament in the year 2002 to preserve the biodiversity. And sharing the resources. That is in the case of the main Biodiversity Act. Mainly it will pledge taken. In the case of Biodiversity Act, in the case of aims to preserve biological diversity, and promote to sustainable use and ensure for sharing benefits from its use. Okay, that is very important. That is the main task or main aim of this Biodiversity Act of India. Biodiversity Act of India in the Parliament in the year 2002. After this global, that is in the case of the World Summit on Sustainable Development conducted in 2002, same year at Johannesburg. That is about this chapter, that is biodiversity and conservation. Okay, we conclude this chapter. Okay, thank you.